Welcome to Part 1 of Lesson 3.2, How Do Normal Cells Become Cancer Cells? The goals of this lesson are to examine how mutations in DNA lead to changes in when and how a protein is made, and compare the consequences of mutations that affect large sections of DNA to mutations that affect single nucleotides. At the end of the lesson, the students should be able to explain how DNA mutations turn normal cells to cancer cells in a process called transformation. Explain the difference between mutations that affect when a gene is transcribed and mutations that affect how a gene is translated. Explain how mutations in proto-oncogenes and tumor suppressors leads to cancer. We will achieve these goals by engaging in a Socratic discussion and a computer lab activity to examine DNA mutations in cancer. To prepare for this lesson, you'll need to review the following key scientific concepts that will be presented throughout this lesson. Our genome consists of both genes and regulatory sequences that control gene transcription. Transcription is the process in which instructions in our genes are used to make a corresponding intermediate molecule, RNA, which is then translated into proteins. Mutations in regulatory sequences may alter when genes are expressed. Normal cells turn into cancer cells by losing control of the genes that regulate cell proliferation and death. This can happen if there are mutations in certain genes or regulatory sequences. This process is called transformation. You can review the scientific content in the background reading provided for you in the teacher primer, the teacher manual, and the student workbook. The teacher primer provides in-depth knowledge about the scientific content presented in this lesson. The teacher manual or lesson plan provides a minute-by-minute explanation of the lesson structure. And the student workbook provides additional explanation for students. Be sure to print the Lesson 3.2 Part 1 worksheet. The key points of the do now are that the first step in transformation of a normal cell into a cancer cell is to lose control of cell proliferation. This occurs by keeping proto-oncogenes switched on and switching tumor suppressor genes off, leading to hyperproliferation. Much like us, our cells go through key stages during their lifespan. We call these stages cell division, signaling, differentiation, aging, and death. Cancer cells lose control of these key stages. This is called transformation. Transformation occurs because of mutations or permanent changes in key genes. Next, we take a closer look at these key genes. When proto-oncogenes are mutated, they become oncogenes and the cell continues to proliferate. When tumor suppressor genes are mutated, the cell loses control over its ability to halt proliferation. You can think of a proto-oncogene as a gas pedal and an oncogene as a gas pedal that is stuck on, whereas a tumor suppressor gene is a brake, and a mutated tumor suppressor gene is a brake that is stuck off. When both are mutated, the cell hyperproliferates. The key points of the lesson's discussion are that mutations to large segments of the genome that affect regulatory sequences can switch genes on and off, even if the gene itself is normal. Mutations within genes themselves that affect amino acid sequence and protein structure can inactivate protein function. Some mutations can switch genes on and off. For example, if transposons that contain promoters jump in front of genes, they can turn the gene on. If a transposon jumps inside a gene, that gene may no longer be able to be transcribed and will be turned off. Mutations can also affect how a gene is translated. For example, if a gene has a mutation that affects changes in the amino acid sequence of the resulting protein, that protein may be inactive. Remember that mutations occur randomly every time a cell replicates its DNA. A cell begins to replicate its DNA by unwinding its chromosomes. At this stage, it is very unstable. If the chromosome breaks and doesn't recombine properly, large segments can rearrange. This can separate genes from their promoters or put them near new promoters. In either case, gene expression will be abnormal. These kinds of rearrangements are called translocation mutations. If they affect genes that regulate cell proliferation, they can start a cell on the path towards transformation. Indeed, translocation mutations are common in leukemias. However, these types of mutations are not the most common type of mutation. In fact, the most common type of mutation occurs when a single nucleotide is miscopied and either omitted or substituted with another nucleotide, for example, a G replacing an A. This kind of mutation is called a single nucleotide polymorphism, abbreviated as SNP. SNPs are important when they cause the amino acid sequence in a protein to change. Make sure students have a solid understanding of the central dogma of biology, which is that proteins are made of amino acids and that cells make proteins based on the instructions in their genes. The DNA sequence for each amino acid in a protein is coded by 3 nucleotide bases. This is called a codon. Each codon contains a different sequence of nucleotides. Each amino acid has several possible codons. A good analogy to help students think about how mutations to a DNA sequence can affect the proteins produced is to divide this DNA sequence into a sentence of 3-letter words, starting from the left. Each letter in the words is a nucleotide. Each word is a codon. In the activity, students will see how rearrangements of this sentence will change its meaning. The key points of the activity are that single nucleotide polymorphisms, or SNPs, are mutations in which a single nucleotide is changed. SNPs that do not change the amino acid will not affect protein structure or function. They are called synonymous. SNPs that do change the amino acid may affect protein structure and function. These are called non-synonymous. Insertions and deletions of single nucleotides are called frameshift mutations and can have severe consequences. Students first complete the Part 1 worksheet that makes them comfortable thinking about mutations to codons. Deleting a nucleotide is called a deletion mutation. Notice how all the words after the deletion do not make sense, and the sentence loses its meaning. Likewise, a deletion mutation will prevent the right amino acids being made, so the protein cannot be produced. Adding an extra nucleotide is called an insertion mutation. Notice that just like in a deletion, the words after the mutation don't make sense and the sentence cannot be read. Likewise, this protein cannot be made. Because deletions and insertions alter how the codons are read from their DNA template, they are called frameshift mutations and are the most severe kinds of mutations. Changing one nucleotide for another is called a substitution mutation. Notice that this affects only one particular codon. There are 2 possible outcomes. The meaning of the sentence may change as it does in the example. This is analogous to protein function being altered. But because there is more than one codon for each amino acid, the amino acid might not change, and in this case, the structure of the protein will not be affected. We call mutations that don't change the amino acid synonymous, and mutations that do change it non-synonymous. This lesson is in 2 parts. Carry on to Part 2 to end this lesson.